Let's talk about something truly amazing. Happening right now, inside every single one of your cells, there's this invisible, high-stakes battle going on. It's a nonstop fight to protect the single most important molecule in your body, your DNA. So today, we're going to meet the microscopic crew that's working 24/7 to keep your genetic blueprint safe and sound. So why does this repair crew matter so much? Well, to really get it, we have to start with this question right here. The answer ties directly to a word we all know and, frankly, fear: cancer. Understanding this connection is absolutely key to seeing why this cellular maintenance isn't just cool science, it's a matter of life and death.
At its core, cancer is really a disease of rebellion. It's when cells just go rogue. They start multiplying like crazy, completely ignoring all the signals from the body telling them to stop. And all this uncontrolled growth is what ends up forming tumors.
So what makes a perfectly normal cell start this rebellion? Well, the problem lies in the cell's instruction manual. It's DNA. The root cause of all this chaos is an accumulation of mutations, and mutations are just permanent, damaging changes to the DNA sequence itself.
Okay, now here is the part that might just blow your mind. DNA damage isn't some rare freak accident. No, it's happening constantly, all the time. As your cells divide, mistakes get made. As you're just sitting there, things from the environment are causing wear and tear. This constant assault is the normal, everyday reality inside your body.
So you might be wondering, if our DNA is under constant attack, how are we even alive? Well, our cells have an incredible first line of defense. These amazing systems designed to catch and fix errors the very second they happen, right in the middle of DNA replication. Think about this first system, proofreading, like a live spell checker for your DNA. The little molecular machines that build new DNA strands, they're called DNA polymerases, are super careful. As they add each new genetic letter, they actually double-check what they just did. If they spot a mistake, they literally back up, snip out the wrong letter, and pop the right one in before moving on. It's just an incredibly efficient process. But, even the best spell checker isn't perfect, right? Every once in a while, a little typo, a mismatched space, slips past that initial proofreading stage. So what's the backup plan? This is where the cell's copy editor comes in, mismatch repair. This system scans the brand new DNA right after it's been made. A whole team of proteins finds the mistake. Then other enzymes swoop in and cut out the wrong section. A DNA polymerase comes in to fill the gap correctly, and then finally, this molecular glue called DNA ligase seals it all up. Good as new. Okay, so that handles errors made while copying. But what about all the damage from the outside world? Things like sunlight, chemicals in the air, radiation. They're constantly attacking our DNA. For that kind of threat, the cell has to deploy a specialized cleanup crew. Now, this cleanup crew has 3 main strategies in its toolkit, and each one is designed for a different type of damage. We're going to look at all 3. A super simple undo button, a precise surgical cut, and a more heavy-duty patch job. Let's break them down. All right, the most elegant fix is called direct reversal. It's pretty much a biological undo button. Seriously, for certain types of chemical damage, a specific enzyme recognizes the problem, like an unwanted chemical tag stuck where it shouldn't be, and it just plucks it right off. It reverses the reaction and restores the DNA. Simple, clean, and super effective. But, of course, not all damage can be undone so easily. For the more complicated problems, the cell has to use a cut-and-paste approach. This is called excision repair, and it's a 2-part strategy for dealing with the tougher stuff. First up, we have base excision repair. You should think of this like a tiny, highly precise surgical operation. A special enzyme scans the DNA looking for one specific type of damaged letter. For instance, sometimes a cytosine base can turn into a uracil, which doesn't belong. The enzyme finds it and snips out just that single bad base. Then the rest of the crew comes in to cut out the empty spot in the backbone, fill it with the right letter, and seal the whole thing up. And that brings us to the next level, nucleotide excision repair. This system is for the big problems, damage that's so bulky it actually twists the DNA double helix out of shape. We're talking about the kind of damage you get from UV rays in a sunburn or from the chemicals in cigarette smoke. The machinery finds this distortion, pries the DNA open, and cuts out a whole patch of the damaged strand. And then, just like before, the builder enzyme and the glue enzyme come in to rebuild and seal the gap.
Okay, now we're getting to the most serious injury that DNA can possibly get. We're not talking about a wrong letter or a twisted patch anymore. We're talking about a full-on, catastrophic break, where an entire chromosome literally snaps in half.
This quote from our source material really just says it all. A double-stranded break is a code red 5-alarm fire for the cell. If this doesn't get fixed, and fast, entire chunks of your genetic code, we're talking hundreds of genes, could just be lost forever.
Now, the cell has two very different ways of dealing with this disaster. On one side, you have the emergency, quick and dirty fix, non-homologous end joining. This is where the cell basically just grabs the two broken ends and shoves them back together as fast as it can. The problem? It's messy. Some DNA letters might get lost or added by mistake, which causes a mutation. But hey, a small mistake is way better than losing a huge piece of a chromosome. On the other side, you have the much more elegant solution, homologous recombination. For this, the cell finds the nearly identical, undamaged copy of that DNA sequence on its sister chromosome and uses it as a perfect template to flawlessly rebuild the broken part. It's slower, yeah, but it's way cleaner and almost never causes a mutation.
So we've seen this incredible, multi-layered defense system that's constantly at work. But what happens when the repair crew itself is faulty? What happens when these amazing, elegant systems actually break down? And here is the absolute crucial point. The repair crew itself is built from instructions in our DNA. So if the genes that are supposed to build these repair proteins are themselves mutated and broken, the cell loses its ability to fix any mistakes. This is the genetic basis for a lot of hereditary cancers, the kinds that can run in families. Let's make this real with two specific examples. In Lynch syndrome, that copy editor system, mismatch repair, is broken. This means typos build up like crazy, which dramatically increases the risk of certain cancers, especially colorectal cancer. Then you have xeroderma pigmentosum. Here, the nucleotide excision repair pathway is faulty. That means the body has no way to fix the damage caused by UV light from the sun. And the consequences of that are just devastating. Without being able to repair sun damage, people with this condition are so incredibly sensitive to sunlight that about half of them will get skin cancer by the age of 10 if they don't take extreme, constant precautions. That one statistic just shows you how absolutely critical these repair systems are for our survival. So just take a second and think about that. Every single day, your cells are performing a number that's almost impossible to imagine. Trillions of these repairs, and most of them are flawless. All just to keep you healthy. It's this constant microscopic ballet of finding damage, cutting it out, and rebuilding. It's more than just a biological process. It's a story of constant vigilance and incredible resilience. And it really makes you wonder, right? What can this amazing built-in system of resilience at the cellular level teach us about facing the challenges in our own lives?